Hello, welcome to Biology, the scientific study of life. Entire genomes can be mapped at the DNA level. As early as 1980, molecular biologist David Botstein and colleagues proposed that the DNA variations reflected in RFLPS could serve as the basis of an extremely detailed map of the entire human genome. Since then, researchers have used such markers in conjunction with the tools and techniques of DNA technology to develop more and more detailed maps of the genomes of a number of species. The most ambitious mapping project to date has been sequencing of the human genome. Officially began as the Human Genome Project in 1990. This effort was largely completed in 23 with the nucleotide sequences of the vast majority of DNA in each human chromosome, the 22 autosomes, and the pair of 6 chromosomes was obtained, organized by an international publicly funded consortium of researchers at universities and research institutes. The project proceeded through 3 stages that provided progressively more detailed views of the human genome: genetic or linkage mapping, physical mapping, and DNA sequencing. The interview with Eric Lander on page 236 to page 237 gives a personal view of the project. In addition to mapping human DNA, researchers with the Human Genome Project are also working in the genomes of the other species important in biological research. They have completed sequences for E. coli and numerous other prokaryotes, such as Saccharomyces cerevisiae yeast and Cynorhybris elegans nematode, Drosophila melanogaster fruit fly, and moss muscular mouse, and quite a few others. These genomes are of great interest in their own right and are also providing important insights of genobiological significance. As we'll discuss later, in addition, they are early mapping efforts on these genomes were useful for developing their strategies, methods, and new technologies necessary for deciphering the human genome, which is much larger. Genetic linkage mapping, relative ordering of markers. Even before the Human Genome Project began, earlier research had painted a rough picture of the organization of the genomes of many organisms. For instance, the karyotype of a species reveals the number of chromosomes and their overall banding pattern. Notice this figure. And some genes had already been located on a particular region of a whole chromosome by fluorescence in situ hybridization, FISH or FICH, a method in which fluorescently labeled probes are allowed to hybridize to an immobilized array of whole chromosomes. Notice this figure. Cytogenetic maps based on this type of information provided the starting point for more detailed mapping. With cytogenetic maps of the chromosomes in hand, the initial step in mapping a large genome is to construct a linkage map of several thousand genetic markers spaced throughout each of the chromosomes. Stage 1, notice this began cytogenic map. Stage 1, notice that. Then notice the order of the markers and the relative distances between them on such a map are based on recombination frequencies. The markers can be genes or any other identifiable sequences in the DNA, such as RFLPS or the sample sequence DNA. A primary on sample sequence DNA, which is abundant in the human genome and has various alleles differing in length. Researchers compiled a human genetic map with some 5,000 markers. Such a map enabled them to locate other markers, including genes, by testing for genetic linkage to the now known markers. It was also available as a framework for organizing more detailed maps of particular regions. Singular or physical mapping, ordering DNA fragments. In a physical map, the distance between markers are expressed in some physical measure, usually the number of base pair along the DNA. For a whole genome mapping, a physical map is made by cutting the DNA of each chromosome into a number of restriction fragments and then determining the original order of the fragments in the chromosomal DNA. The key is to make fragments that overlap and then use probes or automated nucleotide sequencing of the ends to find the overlaps. Stage number 2, notice that this one and this is stage number 2. Physical mapping. In this way, more and more fragments can be assigned to a sequential order that corresponds to the order in a chromosome. Supplies of the DNA fragments used for physical mapping are prepared by cloning and working with large genomes. Researchers carry out several rounds of DNA cutting, cloning, and physical mapping. The first cloning vector is often a yeast artificial chromosome, YAC, notice that, which can carry inserted fragments, a million base pair long, or bacterial artificial chromosome, BAC, an artificial version of bacterial chromosome that can carry inserts of 100,000 to 500,000 base pair. After such long fragments are ordered, each fragment is cut into smaller pieces, which are cloned in plasmids or phages ordered in turn. Notice that, and finally sequenced. DNA sequencing ultimate goal in mapping. The ultimate goal in mapping a genome is to determine the complete nucleotide sequence of each chromosome. Notice this figure also. Stage 3. If a pure preparation of many copies of a DNA fragment up to about 800 base pair in length is available, the sequence of the fragment can be determined by a sequencing machine. The usual sequencing technique described in this figure was developed by British scientist Frederick Singer, often called the dideoxyribonucleotide or the dideoxy for short term chain termination method. Even with automation, the sequencing of all 2.9 billion base pair in a haploid set of human chromosomes presented a formidable challenge. In fact, as discussed in the interview on page 236 and 237, a major thrust of a human genome project was the development of technology of faster sequencing, along with more sophisticated computer software for analyzing and assembling the partial sequences. In practice, the 3 stages shown in this figure overlap. Notice that. Also overlap in a way that our simplified version does not portray. But they accurately represent by overarching strategy employed in the Human Genome Project in 1992, emboldened by advances in sequencing and computer technology. Molecular biologists G. Craig Venter devised an alternative approach to the sequencing of whole genomes. His idea was essentially to skip the genetic mapping and physical mapping stages and start directly with the sequencing of random DNA fragments. Powerful computer programs would then assemble the resulting very large number of overlapping short sequences into a single continuous sequence. Despite this skepticism of many scientists, the value of Venter's approach became clear in 1995 when he and colleagues reported the first complete genome sequencing of an organism, the bacterium Haemophilus influenza, in May 1998. He set up a company, Cellular Genomics, and promised a completed human sequences in 3 years. His whole genome shotgun approach was further validated in March in March 21 with completion of Drosophila melanogaster genome sequence as promised in February 1921. Cellular announced the sequencing of over 90% of the human genome simultaneously, with similar announcement by the public Human Genome Project. Representatives of the public consortium point out that Cellular made much use of consortium's map and sequence data, which are immediately made freely available to all researchers. Unlike Cellular's data, they further assert that infrastructure established by their approach greatly facilitated Cellular's effort. Venter, on the other hand, notice this. On one level, genome sequences of a human and other organisms are simply a list of nucleotide bases, millions of whole genome. Shotgun approach, the sequencing. This approach, developed by Cellular Genomics, random DNA fragments are sequenced and then ordered relative to each other. Notice this one. Cut the DNA from many copies of an entire chromosome into overlapping fragments short enough for sequencing. Notice this stage 1. Clone of the fragments in plasmid or phage vector. Notice this. And stage 3, sequence each fragment, and 4, order the sequences into one overall sequence with computer software. Notice also back to 3-stage approach to mapping an entire genome, starting with a cytogenetic map of each chromosome. Researchers with the Human Genome Project proceeded through 3 stages of mapping the reach of ultimate goal, nearly complete nucleotide sequence of every chromosome.